one. Point. Yeah, we want to get a better one. What do you think?
Other options? Laser point.
Pick one? That's one.
That's the perfect choice. This is the one you use, right? Yes.
Okay. Yep.
But I hope it does.
People
The
are
morning you start, tell them we're about to start tonight. You guess the morning. Like say we're about to start.
about to start.
This is good.
I'm not sure if it's going to
On neck, head, back of the head, back of the head. She's saying put in the back of the head. No, not so.
Okay. This is for the ear.
Oh, we've been putting it off for a long time.
Yeah. Yes. Good.
Thank you.
Okay.
Just yeah. It's fine. These pointer options. Laser point. That's the
Okay.
Thank you.
Good. I think this is a bad spoon. Okay. All right, I'll just use this. I don't know how to do this, but can you hear me okay? All right. So much for the point. Can't get it to work. Can you see this okay? All right. But basically, don't panic. I think you can tell it's going to be a pretty complicated little session. But I've opened up a session Friday morning. So that if we need, we're just going to keep going, okay? No need to panic. But in terms of the secretory function of the stomach, you can divide it functionally into two parts. The Sst gland area, which, okay. The Sst gland area, which includes the fundus and the, am I losing it? I can't get one to you. Okay. The Sst gland area. Did that go away for you? Yeah, it's going away for me too. I have a feeling this experiment's not working out very well. I can keep trying. Okay, if it doesn't work, it doesn't work. Okay. The eccentric gland area, which is about 80%, and anatomically the fundus in the body, and it's your acid producing portion of the stomach. And the pyloric gland area in the antrum primarily, and it's your gastrin producing portion of the stomach. Gastrin is the hormone, and circulation, come back, and affect the synaptic gland area to secrete acid. Okay, we'll come back to it. All right, this is what I call functional mucosal anatomy. And what we're driving at here is this is the synaptic gland, this is the pyloric gland area, and we have surface mucus cells here, and some mucus producing cells in gastric pits as well. But what I'm trying to attract attention to the stem cell here. They're in the progenitor zone. It's in the isthmus, okay, it's a little lower in the pyloric area. But these stem cells are pluripotent cells. They continuously divide, and then they migrate either up or down and differentiate, mature, okay, into the other cells that populate the epithelium, okay? So in terms of the mucus surface cells, they migrate up. Here in the pylorus, a lot more mucus producing cells. They migrate up, and then they're shed. Okay, it's called the noises. You don't have to know that they die and they slough off. Okay, so you have a lot of dead cells, but they're always being replaced. So these stem cells are replacing all the cells that are being lost, and basically it's referred to as turnover. The turnover in humans in the GI tract is anywhere from 3 to 5 days. The cells in the pit, like the parietal cells that make acid, you see that nice shape, kind of aggressive shape. Okay, I'll get into that in a minute. And you have Chi cells that make carcinogen, things like that. They take a little longer to turn over and basal part of the gland. And now we're going to be dealing with the I mentioned gastric, but actually the most important part is gastrin histamine axis. How does that work? Well, gastrin, as I mentioned, released by the pyloric gland into the blood. It's a hormone. It goes around, comes by, and doesn't, very little of it directly affects the parietal cell. We'll get to that later. The most important stimulus for acid secretion is through this pathway where the gastrin comes up in circulation, impacts on enterochromatin-like cells, which all of you know. And you notice these they're not open cells, they're closed, so they have to come up in the blood. And these cells release histamine. Histamine works in a paracrine fashion. To stimulate acid secretion. You can see that better here, where we have a histamine release right next to the parietal cell. Okay, that's called gastrin histamine axis. Here, we're going to get involved with inhibitory factors. Mentioned somatostatin a long time ago, I think. It's going to be an inhibitor, no matter where you run across it. And basically, it can impact all the components of this gastrin histamine axis. Okay, your D cells are the ones that make somatostatin, and you notice if you drew a cartoon here, they have extensions actually on the BCL cell, which make histamine. They have extensions on a parietal cell, and in the pyloric area, somatostatin will have extensions on gastrin secreting cell. Okay, so what you've got is you're inhibiting this entire pathway by somatostatin being present in both the extrinsic and the pyloric area. So far, so good? We're good. Okay, so stimulatory factor, inhibitory, somatostatin. The last note will come up later, not that important here. Oh, yeah. Enterochromatin cell. You notice that the ECL cells, they stand for enterochromatin-like cells. These are the enterochromatin cells. Don't ask me how these names happen. I assume this one was found first, and then someone saw these and said, well, that's enterochromatin-like. Okay. But anyway, ECL cells are not the same thing as EC cells. Okay. One makes histamine, the other one makes 5-hydroxytryptamine or serotonin. think we'll get to that later. All right, this slide lists the major constituents of gastric juice, okay? The only indispensable part of gastric secretion is an intrinsic factor, if you will. It's indispensable because you need it in order to absorb vitamin B12. Without it, you don't absorb vitamin B12 very well, okay? So it's the only people who don't have a stomach that can have it. They need injections of vitamin B12. But as far as nutrition or anything goes, not a problem. Okay, so this is the only indispensable factor. Okay, now in terms of digestive function, I wish I didn't do that, but okay, you have hydrochloric acid. And hydrochloric acid is important in converting pepsinogen to pepsin. We'll cover that in a minute. But its direct contributions to protein digestion is simply denaturing the ingested protein, okay? We'll come back to that. And you have pepsin, obviously it's going to attack peptide, right? Or protein, if you will. It's all hydrolysis, by the way. Pepsin is an endopeptidase. We'll cover that again a little bit later. That means it'll cleave peptide bound with the internal, not on the terminal end of the protein or peptide. Chief cells may secrete lipase, and it does have some effect on triglyceride hydrolysis, but neither pepsin nor gastric lipase is really essential. You have pancreatic enzyme, which we'll cover, I think, at the end of this week, I guess. And those are the most important enzymes for processing carbohydrates, lipids, and protein. The other thing that I'll try to attention to, the protective function of gastric juice. Hydrochloric acid, it kills bacteria. But what I have is impede bacterial colonization. Bacteriostatic, this simply means to stop them from replicating, and silent means to kill. Okay, pepsin, the same thing. It will attack protein in bacteria cell wall. Okay, and it's impede bacterial mucus, decrease bicarb. It's a chemical barrier against acid, so it's protective, but it also impedes bacterial colonization. Are you getting the general drift here? It seems like you can remove the stomach, or a person can have achlorhydria, no acid secretion, and he's fine, but he's going to be more prone to infection. One of the primary things the stomach does is sterilize incoming material. Remember, your salivary glands do the same thing, okay? And of course, if you detected something that wasn't very nice, you spit it out, so your defenses started right from the beginning. This is a cartoon of a varietal cell. This is resting. I'm sure you've got this in histology. It's a cartoon. But hydrogen potassium ATPase, that's your proton pump. And I think in the TBL today, you were talking about proton pump inhibitors. They specifically inhibit this ATPase. Okay. But at any rate, this hydrogen potassium ATPase is these membrane, the tubular membrane, compact structure, and there's little openings, or cisterna, or canaliculi, and if once you activate it and make it secrete maximally, the entire morphology changes. These tubular vesicles fuse with the canaliculus, the fusion, and microvilli show up, and you have this huge surface area where all your proton pumps are located. Makes sense? And of course, this is open to the lumen, it's making acid, but this is a tremendous increase in surface area by the microvilli. Okay, so the whole morphology changes, and when you turn off acid, it goes back to looking like this, histologically. Okay, but that's why you see that cartoon of an aggressive varietal. Every time you see this, you know they're talking about a varietal, but that's where it came from. It's just morphological circulation. All right, so here's your proton pump, and what I'm showing here is stimulatory factor and inhibitory factors. Now, the stimulatory factor, gastrin, acetylcholine, gastrin, you know, we're working on cholecystokinin 2, or some textbooks say CCKB. Okay, it's different than CCKA or CCK1, but that's because gastrin and cholecystokinin have identical terminal amino acid. If you remember the first one, you remember that part, but it's called CCK2 in the parietal cell. Muscarinic receptor and H2 histamine, as opposed to the other ones made by mast cells involved in allergic reactions. That's histamine too, but it's not H2. Okay, that's it. Now, the potency of these factors on the parietal cell, histamine is most potent. Okay, followed by acetylcholine, and the least potent is gastrin. Okay, that's why the gastrin histamine axis is very important. We'll come back to that in a minute. Now, we've covered somatostatin already. We know it's inhibitory, right? We'll be dealing with prostaglandin probably after you come, no, this week sometime, I think. Yeah, would be, but they're inhibitory as well. And here in the parietal cell, these gastrin and acetylcholine will stimulate calcium. That's the cell signal they use to promote this morphological gel change and acid production, whereas histamine works through cyclic AMP. And as I mentioned, histamine is the most potent. Also, guess where the inhibitory factors come in? On cyclic AMP, not the thing, but histamine activates, right? The second messenger of histamine activates. All right, and here I have potassium channel, chloride channel. Well, hydropotassium ATPase, it's like sodium potassium. Okay? It's putting out a proton and exchanging it for potassium. So you need potassium. So this potassium channel serves for recycling of potassium. You knock out this channel, you can't recycle potassium. Eventually, this pump stops. You need to prime it with something. You need the potassium, right? Now, chloride, that's why you need chloride. The proton or the hydrogen is going to join with the chloride to make hydrochloric acid. So you need a chloride channel. And people pretty much think this is your CFTR. Remember that? Cystic fibrosis tract. Remember? CFTR. Okay. Now, you know I'm going to be talking about transporters, right, eventually. I know you hate this stuff, but this isn't too bad, okay? You've already gotten exposed to most of this, and we just talked about it pretty much. I'm not going to talk about this, that the sodium ATPase, right? So let's take a look at this one. They're all labeled here. Where's one right here? Carbonic anhydrous, you know what that is, right? It takes carbon dioxide and water, forms carbonic acid, and that dissociates. Now, the hydrogen potassium ATPase picks up the proton, okay, and exchanges it for potassium. Okay, so you need this potassium channel to keep recycling it, as I mentioned, potassium, and you're putting out the proton here. Now, the cell is not going to like acidification or alkalinization. Okay, there's no way it's going to survive. So once you're moving the proton in one direction, guess what you have to do? You get rid of the bicarb, right? And what's your favorite way of moving bicarb? Exchanging it for another anion, right? When you're exchanging here, it's an anion for an anion. We'll get to that later. But actually, this is called anion exchange. If you move bicarb into the blood side, blood here, and chloride. Now, chloride can be used by this chloride channel, CFTR, okay? But also, anytime you need to move a lot of chloride, and we're making a lot of acid, you need a lot of chloride, Anytime you do that, you need this co-transport. Now, co-transport is going to try to stay electroneutral, right? So you're moving one sodium, one potassium, but you're moving 2 chloride. Is that okay? We covered it before. And that's all there is to know. Now, the extent of acid production by the parietal cell stomach is reflected in the blood cell. The venous blood draining the stomach is active. There's so much bicarbonate put into the blood because of this acid secretion that the pH goes up gastric in this blood. Okay? And it's called an alkaline tide. That's the measurement. Is that okay? That's easy for transporters, right? Now, aren't you glad I tortured you during the salivary lecture? This is not so hard anymore. And you'll see it get progressively more easy. Now, we're going to be talking about acid secretion, but I'm going to bring up pepsin here and mucus in the next slide, I think. But pepsin, there's 2 things to write. It's a nice little cartoon here. Chief cell made pepsinogen, right? It's zymogen, not active. And parietal cells make acid, which is really not, because pepsinogen, in the presence of acid, is called autocatalytic activation or conversion to the active form of pepsin. And basically, it deals with acid. As you know, you have hydrogen bonds and tertiary structures. Well, there's a problem here with the tertiary structure of pepsinogen, okay? Once it's exposed to acid, the active site cleaves off a little peptide that's put in front of it, and now it's totally active. The active site is over. And this pepsin comes back and activates more pepsinogen. So what you've done in the presence of acid, you've moved an inactive peptidase to an active one, and this one causes this cascade of pepsin activation. Does that make sense? So it's a nice little system, all right? We're making parietal cells, making acid, and chief cells making the pepsinogen. Pepsin is an endopeptidase, and here we show you how acid works in pepsin, naive protein. Acid, as you know, it doesn't destroy the peptide bond, right? It just denatures protein, right? You knew that from biochem, right? So basically, it opens it up, which allows the pepsin access to internal peptide bond. That's what it wants to adapt. It's a peptidase. So once the protein denatured, you notice here they're only showing smaller peptide fragments, no amino acid here, because an endopeptidase will not generate amino acid. It stops at dipeptide. Right? There's no internal peptide that binds anymore. The other cute thing is that in the stomach, the pH is low. That happens to be the pH optimum for pepsin. What a beautiful little system, huh? Just showing for comparison, it's trypsin, pancreatic enzyme. It's discharging the duodenum or duodenum, and it's got a higher pH optimum. The reason I'm bringing it up now is the same stimuli that make acid also pepsinogen from chief cells, okay? So acetylcholine can do gastric, histamine, they stimulate. Interesting. So metastatic, that's what it does. Anytime you see metastatic, you know it's going to be inhibitory, right? So it inhibits chief cells, okay, from making pepsinogen. So every time you have acid secretion, more than likely you're going to have pepsinogen secretion. Okay? That's something went wrong. Mucus. The only reason I have this nice cute picture. These are mucus, whatever color that is, orange or something. But that's the mucus being secreted in the stomach. And it's the same thing. Major stimuli for mucus, acetylcholine, histamine. Okay. So when you get acid secretion, guess what? You're going to get mucus secretion too. Okay. So this allows a protective film to develop, or a mucus layer to develop on the gastric surface. And that's shown here in the electron micrograph, EM. This is a gastric fix. I don't know if you've ever seen this, but These are the gastric pits over here where the acid comes out. Stimulated histamine, this whole area is covered with mucus. Okay, these are the asterisks there. All right, so we're going to be talking about gastric acid secretion, but you've got to keep in mind that we're going to be talking also what's going to happen with pepsin secretion going on, pepsinogen, and also mucus. Okay. We're going to be talking about between meals. And then remember salivary secretion, we have cephalic, right? Mediated. Well, here, because it's in the stomach, you're going to have cephalic phase. You can have a gastric phase. And all of that tells you cephalic phase, the stimulus is coming from the brain. In the gastric phase, the stimulus is coming from the stomach. And in intestinal phase, guess what? The stimulus is coming from the intestine. We're going to look at this. And basically, this argues against memorization, right? Nobody wants to memorize it, right? I don't remember. But let's try to figure it out. But let's take a look. We're going to be talking about neuroendocrine and paraendocrine factors that regulate this. And also, you're going to have luminal factors in the gastric phase, okay? So let's take this apart. What do we know so far that we can talk about anyway? Well, we know the gastrin histamine axis. Is that right? Okay. We have the central nervous system here, we have the vagus, okay? It's called vagal tone. I think we talked about that before. It's all ongoing activity, all right? So now the vagus is going to, down in the front, is going to come down, and we know what it's going to release, right? Acetylcholine, is that right? Okay. Acetylcholine, we know what it does with varietal cells, is going to increase secretion, right? In the antrum, the vagus comes down. This area here, the entire nervous system inside the wall. Okay, and here it releases something called gastrin releasing peptide. I wonder what it does. You know, so you don't have to memorize too much here, right? GRP, right? Gastrin releasing peptide. Right? Gastrin, we know what it's going to do. It goes into the blood. Maybe a little bit will actually deprive itself, but the most important thing is it's going to come down here to the enterochromatin-like cell, which makes histamine, which interacts with it, and you have parietal. So far, so good, right? We already knew this, okay? Right? It's all outlined here and in the notes. So this is the most important pathway, and I didn't draw this, so it's not just me making it. I do some other work, but the thickness of this arrow already tells you this is a major play, gastrin histamine axis. Okay. And when I do different colors, I'm trying to attract attention. You know. Guess what? Let's bring in somatostatin. We know it's inhibitory, right? Okay. So to this diagram, we're going to add the cells that make somatostatin. So there's a D cell, right? Okay, so far it's not that complicated, right? Okay, so D cell release somatostatin. It's going to inhibit G cell. So you turn off the gastrohistamine axis, right? And you also have them in the fungus. So somatostatin will inhibit parietal cells directly. We know that. It's also going to inhibit enterochromatin-like cells, so you don't have histamine. So it inhibits all of this gastrohistamine axis. Okay, that's somatostatin. All right, so if somatostatin is inhibiting everything, well, how do we get acid secretion going on, even in the basic state? Well, I don't know, but if you've driven a car, I think most people have around here, you have an accelerator and you have a brake. Isn't that right? An accelerator makes you move fast, but we'll call it stimulatory. A brake kind of inhibits that speed. Okay, you know how these things work. So all we're doing here is playing around with the accelerator and the brake, or the stimulatory factor and the inhibitory factor. But this picture doesn't tell us that. So basal secretion, basically tonic, a vagal tone. And the vagus is active, what's going on here, all I've done is add this pathway. Acetylcholine inhibits somatostatin. Okay, so this, when the vagus comes down, activates GRP and does this, it's also coming down here, turning off somatostatin. So basically, you're putting on the accelerator, letting go of the brake. That's all you're doing. Does this make sense? Exactly, pretty much the same thing happens here. Acetylcholine may be stimulatory of thrive, is also inhibited. Does this make sense now? Is this getting better? Okay, we know how to do this, right? So basically, we have acid. Just because you have your foot on the accelerator a little bit harder than you have on the brake. Okay. So this was a nice little question at the bottom. What's acid from just going on? Well, let's bring in some luminal factors. Now this is a little complicated, but not too much. One more thing added, and that is it's called negative feedback, the Guytonian thing. Very famous for negative feedback mechanism. But once the antrum, this is the antral area, once the antrum becomes acidified, this acid being made in a synthetic gland, which is the fundus corpus, acid is coming down to the antrum, right? Once it starts going through the mucus and the acidic those cells there, it gets turned off. And the way it does that, it activates sensory neurons. And it just happens that these CGR calcitonin gene-related peptides, that's what it's called. I have one right here. There you go, CGRP neuron. And we'll come back to this later in the week, so don't worry about it. It's a sensory reflex. That's what it does. It stimulates, puts on the brakes. It stimulates. So basically, you've got all these systems working. An accelerator and a brake. And luminal acidity will dictate, well, I don't have enough acid, I need more, or I have too much, turn it off. Okay, it's that simple. So we have, it's called SST restraint. The reason it's called SST restraint, we'll come back to this again in the lecture, is because if you knock out SST, okay, you raise you raise basal acid secretion 5 different times. If you don't have your foot on the brake, you're going to be going past the speed limit. Okay, that's all they're trying to tell you. So it's called SST dominant phase, interdigestive basal secretion. Basically, what you end up with is low volume, low pH. You got acid coming out, be in control, but it's going to maintain a low pH in the stomach. By the way, I don't have the time here, so let me know when we'll make up. All right. Now, during the cephalic phase, you have smell, taste, right? Guess what? All you're doing is the vagus system slams down on the whole thing. Well, what do you think's going to happen? You're going to be turning on acetylcholine to provide GRP, turning off the brakes, SST, right? You're turning it off. So basically, you're already making a lot more acid than you were before, just because the vagus got turned on more than it was. It's already tonically activated, but now sight, smell, taste, things like that will turn it on maybe. So it alters the balance that we say down here of the inhibitory and stimulatory factors to the stimulatory. Okay, so it's moving towards stimulation. The gastric phase deals with most of it, 60% of your acid secretion. And here, let's go 1, 2, 3. 1, you have distension. When food's coming in, it's going to deform the epithelium. Then, you know what I mean? That's going to be a next lecture. And it's going to stimulate then the chromatin cells to release, well, 5-hydroxytryptamine or serotonin. That goes up, turns on the vagus. It's fun to do it, it's turning on the vagus again, okay? So, and that's why I have this arrow here, the vagus is up. Peptides are going to be detected by G cells. They have an open configuration, so their apical ends are open to the lumen that detect peptide, and it stimulates gastric secretion, okay? And now the food that you eat almost invariably results in buffering of the existing acid. So the pH actually starts going up. In other words, the pH during ingestion of a meal, once the food hits the stomach, the pH starts going up. It can reach as high as 5, pH 5. Okay. And so what you've done is you've turned off this particular sensory neuron that responds to acid in the room. So far, so good. Well, we're done. All you have to do is just keep going over, okay? Intestinal phase, basically you're returning back. Sorry, did something go on? Can you still hear me? Yeah. The intestinal phase basically takes you back, okay? You lose your sensory cues. You're not tasting it anymore or smelling it. It's leaving your stomach. So you have less distension. All of the luminal factors are going away, less peptide. You're going to have less buffering, okay? The pH going SST going, you're inhibiting gastric histamine. You're turning everything on, okay? That effect is you start decreasing your acid secretion, all right? And you start emptying. So now you're becoming more and more back to where you were before. Let me know when it's time for lunch, okay? Anyway, the intestinal phase has a cute trick because consider it protected. Remember, I told you reflexes and things of that nature. If they're feeding backwards, they're going to be inhibiting something. You know what I mean? That intestine is not interested in giving you more. It's telling you, hold on. Okay. And basically, they respond to fats and acid, and lipid and acid. By the way, fatty acids, long chain. Is it time? I don't know what I'm doing wrong. Is that better? I can't win this game. Anyway, we've seen this before. Well, now it works pretty good. Okay, sorry about that. Anyway, long chain fatty acids are a cytotoxin. And we're going to be getting into high density lipoproteins, low density lipoproteins. They're all being transported in the blood bound to something. You don't find through fatty acids because it's toxic. And of course, the acids are no too. Acid's not very good. So these things, if we've got a lot of acid coming into the duodenum, hold on a second, we don't need it anymore, right? Or lipid. And the way that works is fatty acids will stimulate cholecystokinin. That's it. We have that in the first chapter. Now you're kidding. You're not going to have to memorize these things anymore, right? So cholecystokinin, guess what? It's going to activate extrinsic AFRs, vagus. By the way, the cholecystokinin receptor on the sensory nerve is CCK1. You don't have to know that, but it's a different CCK receptor. And it comes back, and it's going to inhibit, it's going to activate SST, the G cell, which makes up acid, is also going to stimulate your C cell that controls the actually component cells. They respond to that. And it's going to release secretion. Also, your S cell in the duodenum respond to that. The ones that release CCK are I cell. Okay. But anyway, it's the same concept. Extrinsic APRs get activated. They come along here and they activate your D cell. And guess what that does? Now, this is another cartoon of the gastrohistamine act. Wow, look at how many arrows we have going from gastric to ECL cell, which make histamine, but only one arrow going to the parietal cell. Again, just reinforcing the fact that gastrohistamine action is a major player. All right? And so what happens is ordinarily, you have gastrin going into the blood, it activates the chromatin light cells like this. That histamine stimulates parietal acid. This acid comes down, okay, turns this down, then you got this regulatory mechanism. All that this does, turn around some more. Okay? So it starts to inhibit acid secretion in the stomach. So acid when we do a duodenum, going to inhibit. Upstream is going to turn it off. It doesn't want it anymore. And it's going to be the same thing with lipids. Okay? Are we too complicated? We're okay so far, right? This is a quick review. And we're done. Was I going too fast? As long as you can catch on, try to go through this. And as I said, if it's a complicated issue, I can go over it again. I open up a slot. I think it's Wednesday morning. But now that we've pretty much covered it, I have a feeling we can sleep in. So we covered it. Quick review. Here we got pH, blue. This is the blue. I wish they used another color. pH is in blue. Acid secretion or production is black here. The dotted line here, okay. And where's the volume? Here. This is the solid black line at the bottom of your stomach. Okay, so the volume is here. pH, acid, just moving around. Acid and pH are here. So before meal, hello? I think it died. That's it. Can I use this now? Hello. Before a meal, I mentioned you're going to have basal acids. Low volume, very little volume, low pH, very acidic, right? Now, when you ingest the meal, you're going to push on the accelerator, right? Get off the brake, and you're going to have acid secretion. Yep, it goes up. See that dotted line is shooting up. And of course, pH is going to go down, right? There's the tricky part. It goes up because you have food. You eat. Food is a buffer. And only when you didn't do that. But see, when you exceed the buffering capacity, that's the only time you're going to start seeing a decrease in pH. Because now food, okay. So far, so good. This is a quick review. Now we move over. Now the meal is leaving your stomach. The volume is going down, right? If it's leaving, it has to go down, right? Because you eat, so it's going down. Acid secretion was going up before, and now the buffering capacity of the food is going down because the food's leaving, right? Acid secretion is still going up. Okay. So now everything comes back toward the base. Okay. And over here also, you have your inhibitory factors coming from the intestine, and that's your acid entering and any lipid you have that might be in there entering, and it's going to slow down acid secretion too. So, okay, you're losing all the stimuli to turn acid secretion on, and you're turning on things that we're putting on. We don't need it anymore. You're processing your meal now. Down here, you're probably already hydrolyzing most of your protein, carbohydrates, and lipids. Okay, but that's it. If you understood that, we're okay. All right.